students. Today we have decided to go for biology chapter. It's genetics and evolution. Remember, this chapter is extremely important with respect to your academic syllabus also. And since you are preparing for the various examinations, NEET 2020 or 21, this chapter has extremely important concepts. It has very heavy weightage in the academic portion also, and your entrance examination. Why we decided to go for directly for the genetics? There is a reason behind it, because if we finished it early, you all will get enough time to revise the notes, to prepare for the exams, and go through the much number of tests. So that becomes very important. So this is very critical and important chapter. Why I'm saying critical? Because it has many basic concepts that is correlated with the 11th standard. So I hope you have studied the 11th standard biology very well, the reproduction part of the biology and the basic concepts of chromosomes, DNA, RNA, the cell division methods like mitosis, and the gametes formation process like meiosis. That will play the extremely important role while studying this chapter. Anyways, in this genetics and evolution, this topic basically consists of 3 chapters. First is just principle of inheritance and variation, and this topic basically consists of or related to only Mendelism. So we know that father of genetics or modern genetics, Gregor Mendel, and he had worked on pea plants. We know about it. In the lowest standard also we studied the inheritance laws of it, and he worked nearly 7 years on the pea plant, that is botanical name is Pisum sativum, and with respect to his observations he had made some laws. That what are the laws, what is the importance while studying the genetics, this we are clearly going to study in this chapter. We'll go very thoroughly this genetics part, because as I indicated first, it is extremely important. Now if you go to the principles of inheritance, it has Mendel's law, then inheritance of one gene, inheritance of two gene, that is multi-allelism. Next, sex determination, that means how can we determine the sex of the particular organism. Mutation, that means how the genetic coding affects the genetic behavior of organism. Genetic disorders means how this probable codings of in DNA of nitrogen bases affects the genetic behavior of any organism. So this is, you can see the first chapter just give the theoretical information about genetic disorders, importance of genetics, and what causes there is a multi-variation among the offsprings than from the parents, and which factors affect the variation in the offsprings. Mendel studied the pea plant according to F1 and F2 generation, that is first filial generations and the second filial generation. That observations also we are going to study in the successive subtopics. So this all is covered in the first chapter. If we go for the molecular basis of inheritance, the name itself you can say molecular. Remember, molecule means they are the biomolecules, and the biomolecules means they are the nucleic acids. Nucleic acids, that is nothing but DNA and RNA, that is deoxyribonucleic acid and the ribonucleic acid. They are the Polymers. So polymers, just overview, we know that polymers means that is a chain of monomers. That means this nucleic acid consists of number of monomers, and that monomers is nothing but called the nucleotides, and the plural form of it is nucleosides. So we'll go this one, no need to worry. This is just, I'm giving the overview of genetics and evolution. That means what are the things we're going to cover in these topics. Then how the genetics works out on the basis of molecular theory. The first topic is DNA and how it affects on genetics. Next is RNA and the types of RNA like mRNA, tRNA and rRNA. What are their basic fundamental functions? How they affect on genetics. Next is replication and transcription. This is the process. Next, genetic code. As I said, the genetic code is nothing but the genetic language of formation of new characteristics in the offsprings. The genetic language, why I'm saying, it's like it depends on the codings of your nitrogen bases, that is the purines and pyrimidines, that is adenine, guanine, thymine and cytosine and uracil. These are the genetic coding effects on the genetism of organisms. Next, Human Genome Project, that means the advancement in the genetics. Next, we have the evolutions, that means from the ancient time to right away. What are the basic fundamentals which resulted in the evolution of the human beings and the multi-organisms and their characteristics, their fundamental evolution in the organisms. So this is all we are going to study in the evolution. The origin of life and its theory, adaptive nature of radiations, biological evolutions mean the changes in the organisms, and Hardy-Weinberg principle, and the origin and evolution of a man. This origin and evolution of man, we had some idea about it, but in this topic we are going to study thoroughly. So from this explanation, you will understand that basically the first topic is give the general theoretical overview of the genetics. Your second topic, it goes actually in the practicalism, that is how this molecular or biomolecules polymers affects on the genetic characters of human beings. And the last one is evolution, that means after going under the consideration of genetics, how this evolved the human beings or organisms over the period of times and thousands of years of times. So this is what the topics we are going to study. This topic is very interesting. Only things we have to clear is the terms and terminologies should be very clear, and you should have the only clear approach to the topic. So what I suggest is give the as much possible time to this to study right away. And if you become perfect now, you will get the most of the time to study for and prepare for the next exams what you're going to appear for. Thank you. So the next is the term is genetics. The genetics has explained two terminologies. One is the inheritance and next one is the variation. Under the title of genetics, these terms look like quite same, but they have differences between them. Inheritance means how the characteristics are transferred from the parents to the offsprings, and who are the responsible for the transfer of characteristics from the parents to the offsprings. The study of all these things we study under the inheritance. So inheritance do not consist of any difference characteristics between the offsprings and parents. So that inheritance is just limited to transfer of characteristics from the parents to the offsprings. But if we look to the variation term, the variation do consist of transmission of characteristics from the parents to the offsprings, and it becomes extremely important to explain why there is some changes between the parents and offsprings, or why there is some changes between the siblings. So if we look to the fundamentals, suppose we take the example of one couple and who are having the two siblings. So inheritance explains the transfer of characteristics from the couple to their two siblings. It do not explains the percentage or degree of characteristics that is transferred from the couples to the siblings. It just linked to the transfer of characteristics. But if we look to the variation of property, it becomes very needful to explain the degree or extent of characteristics that has been transferred from the parents to the offspring. That means there is a clear indication that all the siblings are not same. And if the siblings are same and they have much of the properties of their parents, so we can say variation is less. That means most of the characteristics has been transferred from their parents to the siblings. As we see in the practical purpose in between the twins, so twins has much of the similarities between them, and that is also related to the inheritance and the variation. So overall, genetics is the study of fundamentals between the inheritance and variation, and this inheritance and variation are closely related to the nucleic acids and the coding between the nucleic acids. That is the bonding between your nitrogen bases adenine, guanine, thymine, cytosine, and uracil. Uracil do not consist in the DNA. It is a part of RNA. It is a pyrimidine nitrogen basis that will go nicely on it in the second chapter, that is molecular genetics. In the first one, we are just simply related to the overview and theoretical part of the genetics. So I hope you people understood what is genetics. Genetics is the study of variation, principle of variation and inheritance, and the characteristics which makes them it to happen. So next is now this study of genetics was well acknowledged by the Mendel, Gregor Johann Mendel, and this mathematician come biologist we can call as the father of modern genetics or genetics. He was born in 1822 and died on 1884, 6th January. This fundamental is not required for your academic portion, but there are some chances they may ask questions in the entrance examination with respect to head, because these all fathers are very important. We should remember all these things. Actually, this Mendel worked 7 years on the pea plant to study the genetics, variation, and inheritance or with respect to this plant and its generations F1 and F2, that we'll study in the successive lectures. His work for 7 years was completely unrecognized. There were some reasons why his work was not recognized well in the contemporary periods and all, but we'll not go into that. So his work came into the recognition after the 16 years of his death. That means he died in 1884, and in 1900 the three scientists made this work to recognize and to publish, and showed the importance of his studies about genetics and his importance of his experiments on the Rhizome sativum, that is pea plant we can call it. The scientists' names are Device, Currens, and Eric von Teckmark. So these are the scientists who rediscovered the Mendel's works in 1900. I have given certain points. So while making the notes, you can prefer these points, and if you want, you can even add some points in it. So basically, Mendel was not biologist. Sorry, he was a mathematician. So his analyzing power or statistical power was good, but that property of Mendel had become biggest problem in his discovery and acknowledging his work, that we'll see in the next subtopic. In the previous subtopics I explained, he was a mathematician, Austrian-born mathematician, and but since he was a priest and he had worked for 7 years on the genetics characteristics of the Bison sativum, the SPLE plant, we'll study why his work was not recognized and what are the factors that made him not to publish his work. There are certain things because his work was not published in the national or international journals. Even as he was mathematician, most of the biologists in the contemporary periods were did not accepted his work. They were not believed on him. Like this can be happen. So these things were not very clear to him, because when they were seeing towards him, they perceive him as a mathematician. So that is also one problem was there. Even the contemporary Darwin theory was quite famous. So everyone, contemporary biologists, were interested in discussing the Darwinism, that is theory of evolution. That also the one more reason that they sidelined the theory of genetics by the Mendel. The next important point is because the study of microscopic genetics or microscopic organisms or microscopic properties or such as genetics and nucleic acids and nucleotides and all kind of things was not most popular, and the factors what Mendel was considered is responsible for the genetics, that was quite not understood by the contemporary biologists. So these all were the reasons why this theory was not heavily accepted or not even accepted by the biologists. These are the reasons for the non-recognition of Mendelism theory after working so hard for 7 years. But if you look to the another part, there are certain points which made the Mendelism theory successful. And this reason where the work what he has done on genetics works were done on this pea plant. That plant has several contrasting characters. The Mendel has selected 7 contrasting characters to study the genetic properties of the Rhizome sativum, and that we will see what are the contrasting characters, how he affected the genetics, and how this contrasting characters helps for Mendel to study the genetic characteristics of this pea plant successively in the successful manner. Second point was the sample size to study this genetic properties was very large amount. So it was become very easy for him to make the conclusion on every point, because if the sample size is very large, then we can conclude the points with great results. So large sample size also was the important point that Mendel has made. So he concluded the points with more clear idea. Next one, since he was originally a mathematician, so his data analysis and his calculations were very correct. So that also gave him a little upper head in calculating the properties of the genetics about the plant. Now, after studying all these things about the pea plant, one more question might be why he selected the pea plant only. What is the reason behind it? So there are also certain academic question they may ask you, what are the reason why Mendel selected only pea plant? Why not others? Because the basic point is it has short lifetime. It has short lifetime. That means several generations can be studied within a short period of time. So next point, what is it can be cross-fertilized or self-fertilized, because pea plant flowers are the bisexual flowers. Even they have the male and female sexual organs. So they are bisexual flowers. Even the self-pollination and the cross-pollination is possible. That is also one advantage the pea plant had. Next is large amount of contrasting characters. So that provided the good clarity on the points and the properties or characteristics. And one more thing is very important is pure line. Pure line, pure line, name itself indicates the pure line. That means there is no heterozygous activity or not mixing of characteristics. This we'll study in the next subtopic. But since this pea plant is basically or predominantly a self-pollinating plant, so it was easy to get the pure line for several generations. So these are the characteristics the pea plant had. So it was obvious choice for Mendel to select this plant and to go ahead for the studying the genetic characteristics of the pea plant. So as we studied, there are numerous contrasting characters this pea plant has. Basically the 7 contrasting characters Mendel had selected to study the properties of genetics about this pea plant. These 7 characteristics you should remember forever, because these are kind of assumptions he made, assumptions about who is the dominant characteristics and which one is the recessive characteristics. So first character he gave is height. In height also we have 2 options, tall or dwarf. So there is no confusion, one is tall and one is dwarf, so it is quite contrasting. So he assumed tall one is a dominant and dwarf or short one is a recessive, and dominant characteristics tall one is capital T and recessive is the small t. So this recessive will follow the small letters and even dominant will follow the capital letters. Next is flower position. Flower position, it can be axillary or terminal. So what is axillary and terminal, I have given the position of flowers for your better understanding, which one is the auxiliary flower, which one is the terminal flower. So flower position axillary was a dominant and flower position terminal was a recessive characters as we assumed. Next is color of flower. Color of flower again he considered as a purple is a dominant and white is a recessive characteristics. Next was pod shape. Pod shape, it is inflated. I have given a small diagram for your better clarity. These are the seed cells I have shown. Inflated means a clear flattened, and constricted, that means you can see the constrictions here. So these are the constrictions you can see in the pods and the seed cells are shown. So constricted pod shape was the recessive and the inflated pod shape was the dominant. Pod color, if it is a green, it's a dominant, and if it's a yellow, it's a recessive. Sheet shape, it's a round, it's a dominant, and recessive, it's a wrinkle. Next is seed color. The dominant is yellow and recessive is a green. Now one thing I have to clear here is, see, this species is a pea plant, but the species is same, but they have the much differences between the different species of the pea plants, and this is differences is mainly due to the alleles present inside the genes. Now what is this alleles, we'll study in the next subtopic. So it is very important to understand this topic, and if we have to understand the topic very well, we should know the basics of terminologies. They have explained this topic. So first we'll go to the terminologies and what is basically this terminologies means and what is their importance in the process of genetics. We'll study about it. So first of terminology we have is gene. Now many times we studied about the gene, but having the clear idea about the gene is very important, and to know what is gene, it is very important to study about the DNA. Basically we know DNA is a double helical structure with it's a polymer, a biomolecular polymer, and it has monomer unit as nucleotides, and nucleotides again has 3 elements, like we have nitrogen bases, we have pentose sugar, and we have phosphate group. This, whatever the names I am taking, it's not that important with respect to this topic, but when we are studying the second topic of this, molecular genetics, that time we'll revise this all very nicely. But to avoid the confusion, I want to show you what I'm about what I'm talking. So what we can do is, I will tell you what this means. Now this is what I'm talking about. So this is DNA, and that means this is not a DNA, it's a monomer of DNA, and this is a monomer of RNA. This is pentose sugar, this is a base, that is nitrogen basis, and this is a phosphate group, and this is again RNA, ribose nucleic acid. This is again a pentose sugar, that is called the ribose sugar, and the sugar which is present in DNA, it is called the deoxyribose sugar. Again, it consists of bases. Remember, the nitrogen bases present in DNA and RNA are different. RNA consists of uracil, DNA does not consist of uracil, and the monomer unit of DNA is nucleotide, and this is same again. So this is just brief overviews that you should get idea at least what is this DNA and what are the element and what are the monomers I'm talking about. So only for that reason I have given this basic structure of these elements, this monomers of this both. So if you add this monomer successively in the number of times, I will get the DNA, and even if I add this monomer in successively times, I will get the RNA. So this is what the basic of this topic. As I said, nitrogen bases, there are 2 nitrogen bases basically we can compare. First is pyrimidines, we can say, this is one more nitrogen base, and next is the purines. So uracil, thymine and cytosine, these are the pyrimidines, and adenine and guanine, these are the purines, and these are the nitrogen bases, and the sequence of these nitrogen bases are basically important, or basically affects on the genetic properties of parents to offsprings, and how these properties are transferred from the parents to offsprings, and why there is a difference or variation between the offsprings itself. So this is all we'll study very thoroughly in the second chapter. So just remember this one. If you can draw the structures while studying the lectures, that will be better. So this is what this different nitrogen bases and the sugar and DNA and RNA. So next one, I will just want to show you what is DNA look like. So this is what the DNA. This is just temporary slides I am giving you, because this is again will come in the second topic. This is nitrogen bases. Again you can see adenine, thymine, cytosine and guanine. This is the nitrogen bases present inside the DNA. This is a double helical structure, we can call this for DNA. And inside this helical structures, you can find out there are certain letters I have written, A, T, and there is a bonding between AT, CG, TA, GC, AT, TA, CG, AT. So what is this AT, CG, AT? That is nothing but called the sequence of nitrogen bases. And the bonding between them, or the sequence between them, suppose if I write the sequence. So first of all, if I write the sequence T, then G, then again T, then A, this is sequence again. Then if you write the sequence, there are a number of different sequences are coming, and the sequences are responsible for the variation in the genetic characteristics between the offsprings from the parents or the sibling themselves. So this is what very important. This will study very nicely in the second chapter. This was temporary one. Now we'll come to the topic, what is gene. So gene consists of definite sequence of your nitrogen bases, adenine, thymine, guanine, cytosine. But remember, your gene is not a DNA, it's a a small stretch of your DNA. It's just small part of it which consists of information, or it codes the information about the genetic characteristics or instruction about it. So it is, these instructions are extremely important for the protein synthesis, and this information only helps the body to produce the proteins, and these proteins, we know that it is the basic building block of life. So genes also very important, but do not confuse with the DNA. It's a small stretch of DNA, it's not a DNA, but the gene consists of a different information, coding information. It's like a coded language for the reproduction, or coded language for genetics, like how we use for binary language for computer applications. Now this production of protein is extremely complex process, and the making of amino acids from the protein itself, it's also again a very complex process. So this we are not going to study these things in this topic. In this topic we are just study how the Mendel properties and the characteristics, and what are the inferences he made from its experiments. Now basically, when Mendel was studying this genetics, he had no idea about the terms called gene. He gave the name as a factors. I told you this, the factors was not known for the contemporary biologists. So that is also one more reason why they discarded the theory of Mendelism. But this factor, what Mendel named, was again reconsidered, but in different name. It's called the gene. That is what we're studying about, is by Johnson. So he is the person, William Johnson. This is a person. He is derived the term as gene. Remember this one, they may ask you. And the term genetics was recognized by the W. Batson. So this is also a different names, scientist names, and their fundamental theories you should remember. Thank you. After studying about genes, the next topic is allele. This is also a very important property. So it is a form of gene. It's quite confusing. Then what is the difference between gene and allele? Remember, if I talk about height, so the height of a person, if I talk about, the gene carries the information about the height, or coding about the height. But the height has again two differences. One will be tall and one will be dwarf. Now this, who decides about the tall and dwarf? And this is nothing but decided by the allele. So height is the basic information coded inside the gene, but the differences between the height as a tall and dwarf, that is by the allele. So allele is a form of gene. It is correct, but it is nothing but it decides the contrasting character between the height. Another one example we study. Suppose I will take the species of frog. I will consider two frogs here. Just assume it. If I consider the species of frog, and now I am considering the property like eye color. So definitely, if I'm talking about eye color, so eye color is coded information inside the gene. That's true. But all the frogs will not have the same eye color. That means there is something which is affecting on the eye color, and that affecting characteristics are affecting property, or the element is called the allele. And this allele makes the difference between the eye color of frogs, suppose as the yellow and the red. So there is a difference between the eye color itself. I told you there is a multiple variations between. These multiple characteristics are derived by the element like allele, or the property like allele. In the previous slide, I gave the example about the couple and their offsprings, and why there is a variation between the sibling themselves, and the transfer of characteristics between the couple and offspring, and this is what the fundamental is. So gene carries the coded information, but the variation between the characteristic itself is by the allele. Next term we have is homozygous. We know all these things. We are very familiar about the homozygous and heterozygous. Homozygous means it is a similarity of the alleles in an organism, or degree of similarity of the alleles in the organisms. This is genetic information. It is height of the plant, if I say. This is homozygous because both are dominant characteristics, so we can call this homozygous, and both are recessive. This is also homozygous. So homozygous means it is the degree of similarity of the alleles in an organism. Next we can talk about the heterozygous plant. It is exactly opposite to this. It is nothing but the different versions of genes. Versions of genes is nothing but the alleles. So each different version of gene is called the alleles. That means if there is a different alleles consist in the specific genes, then we can call this as a heterozygous. If it is different alleles consist in a same gene, it is called the heterozygous genotype. And suppose if I talk about the hair color, we are just discussing about the heterozygous. If I talk about the hair color, if one allele is for red hair, so hair color is a gene. This is nothing but the coded information that is carried by the gene. But again, that coded information has two contrasting characters like red here and brown here. And this red here and brown here are the two different alleles. And whenever there is two different alleles, we can call it collectively as a heterozygous relationship between the alleles. And the relationship between these two different alleles, like suppose I talk about red here is R and small brown here is small r. Now it also affects due to the dominancy of one allele over the other. So we can say it is the red here if R is dominant, or brown here when it is R is the small r is the dominant. So in heterozygous, the dominancy of the particular allele affects on the characteristics or traits. Next is fundamental is a phenotype. We all know him from the tensor is a Punnett square, and that we write the ratio is a phenotypic ratio or genotypic ratio. The basic fundamental of phenotype is just external appearance. We look into the external appearance of the particular plants or particular animals, and we'll write the ratio about it. It is a tall or dwarf or short. So this is external appearance ratio is called the phenotypic ratio. Now I have just taken the genes for height, and genes for height again we have the tall, and this is capital T. Capital T again is tall, and dwarf I can say small t. So the possibilities what I have taken is capital T small t and capital T. Capital this is a homozygous, this is a heterozygous. In Mendel studied the F1 generation, we have all the plants what he observed in they all are tall in the F1 generation. That means capital T is was the dominant characteristics or dominant trait in it. So here also capital T small t, since capital T is dominant, so it will be tall, and capital T again it's a homozygous, so definitely the plant is going to be tall. So this tall and tall, it is just external appearance of the particular element, it is called the phenotypes. But now here this capital T in small t, it is heterozygous. Remember, I'm talking about genes now, different alleles. I'm not talking about the external appearance of the particular element. I'm talking about the genetic element like allele. Here is a capital T and small t. It's a heterozygous, and capital T, it is homozygous. When we talk about exactly the alleles about in and genetic information that is coded by the genes, that is nothing but the genotype makeup, or we can talk genotype ratio. And when we talk about just external appearance, then we can call it as a phenotypic ratio or the phenotypic makeup. Thank you. Next is the next property or next term is the cross. It is nothing but it explains about the plants and reproduction of two plants of different contrasting characters. So just breeding of two plants of different contrasting characters is called the cross. Next is filial generation. The filial means the generation, next generation, like how Mendel observed as F1, F2 and all kind of things. So F1 is the first filial generation, F2 is the second filial generation, and he observed his genetic properties or characteristics of this pea plant, the Sphaerum sativum, to study the different kinds of characteristics of the plant. Next is selfing. Selfing means that is a breeding or reproduction between them, or the pollination between the plants of same generation. If I talk about F1 is the plants which we observed after the first generation. And if I again self them, means there is a reproduction between themselves is nothing but called the selfing. Pure line means this is homozygous plants. And if I do the reproduction between them for 5 or 6 consecutive generations, so I will observe the similar properties or characteristics between them. This pure line is important to get the plants which shows the same type of genotypes. If we don't want the genotype to be different between the plants, then we can observe, or we can do the pure line between them. That can be done by the selfing. Very important topic with respect to your entrance examinations or academics also. It's law of principle or laws of inheritance, we can call this, or more popularly we can call this the Mendel's law of inheritance and variation. So law of dominance is extremely easy. What it explains is, in F1 generation only dominant characters appears. Like suppose if I explain by using this Punnett square, this Punnett name is given by the Ignaz Punnett, and this is a tool to explain or to observe the genotypic ratio and the phenotypic ratio. So if I take this homozygous tall, homozygous tough, these are the same alleles, again same alleles, it is a homozygous gamete, homozygous gamete again. So this is the genetic characteristics. Now if this T and T and this is T and T, so this is explains about the tall, this explains about the dwarf. Now if I pollinate them or by a self or cross, so now TT, this TT, that's capital T small t, the next will be T and this capital T, so it will be TT again. Next is small t capital T, so TT again. Next is small t capital T, then TT again. These are the fundamental outcomes. Now since the contrasting characters in the Mendel observed, this tallness is a dominant characteristics, and according to this law, dominant characteristics always exist in the outcome. So this TT definitely will give the tall plant, this TT will give the tall plant, TT will give the tall plant, and this TT will give the tall plant. So if I want to write the phenotypic ratio of it, all are tall. Whether it is heterozygous or homozygous, we are not worried about it. Just the external appearance, that is all are tall, so ratio will be 4:0. So external appearance ratio is called the phenotypic ratio, and genotypic ratio, that means it is nothing but we are looking to the genes or different alleles present in it. Suppose capital T small t is heterozygous because different alleles, capital T small t different alleles, different and different. So all are heterozygous. So but there is no homozygous property at all. So again all are heterozygous, so we can say it is a 4:0, and all are the heterozygous. In phenotypic ratio also tall was dominant, it existed, and in heterozygous also tall was dominant, it is existed. So law of dominance just explains about the dominant characteristics. The next is second law of Mendel's second law of segregation. Segregation name itself indicate the separation. Now generally every organism we consider as two alleles, but there is also multi-allelism. We will study that in the next few lectures. So it is separation of alleles during the process of meiosis. So first you study the meiosis very nicely, otherwise in the next 1 or 2 lectures we'll study the meiosis. The separation of alleles, and after separating it, each gamete gets one allele. So we know that it is a haploid gamete, and haploid gametes form the diploid zygote. So haploid gametes gets the each separate alleles. That is nothing but called the segregation. Now I have given the example here. Suppose this is a parents and it is 2 alleles are there. The separation of alleles during the meiosis, this meiosis is also called as reductional division, because half number of chromosomes get into the gametes, that's called the haploids, and male and female gametes make the diploid zygote. So during the process of meiosis there is a separation of alleles, means characteristics, and every gamete, when it is a separation happens, every gamete get the separate allele. This is called the law of segregation. Next is law of independent assortment. So this law of independent assortment, the name again itself indicates independency, means they are not dependent on, means alleles are not dependent, individually they can form the gametes. If the two or more gametes alleles are present inside the gene, then they can form the individual gametes independently. So one allele does not influence the gamete formation for the another one. So this is independent assortment, means the individual alleles can form the gametes without any kind of influence on the second allele. So we can law of independent. So we can explain the law of independent assortment by taking this example. So I have taken the dihybrid cross. This dihybrid, monohybrid and trihybrid we'll study in the next subtopic. Just for example, I have discussed here. The dihybrid cross means taking the two properties of pea plant. The tall is dominant, purple is dominant, purple is capital PP. I hybridized with another plant which is short, that is small t, and white, it is small pp. So again two alleles properties. So it is not multi-allelism, it's a two alleles as general we consider. And now if I pollinated them or if I reproduce them, so I can write this, what is the possibilities that allele will form the gametes. So now see here, this capital T and this capital P. So one possibility, otherwise this capital T, another this capital T. So TP, TP for one allele, next TP and TP. So total 4 gametes will be formed. Now you can see here the formation of gamete by this allele is not affected or has no implications on this allele. So they are independent. That is nothing but the law of independent assortment. So this is, I have just drawn this Punnett square, and then now the same thing here. This is what the dominant characteristics gametes, and then here small t, a small p, small t, small p, again this small t and small p, small t and small p. So again 4 gametes will form, and if it is hybridized or pollinized, then this TP, TP, this TP, this TP, so this will form this one. So this is F1 filial generation. We know that in F1 filial generation all the dominant characteristics exist. So here also all the plants whatever is formed is tall and purple, because tall is nothing but the dominant and purple is also dominant. So that should be remembered, because we know that in suppose one in Mendel's experiment, this is a take the example of, this is a tall plant and another plant is a very short. So if they are hybridized or pollinized, so we'll get the F1 filial generation in the ratio we got is 3:1. That means this is TT and this is again TT. Suppose it has, sorry, small TT. We got capital T, small t, capital T, small t, capital T, small t, capital T, small t. That means in F1 filial generation all the plants will be tall. For the diagram nicely, you can just prefer the NCERT book they have given. So in F1 filial generation we can say dominant characteristics always exist. So here also in F1 dominant characteristics was tall and purple. So tall and purple, so all plants, that's 16 plants, if you say properly, this 4 and this 4, 16. So 16 outcomes will be the dominant, that's tall and purple. So we can easily see the gamete formation by this allele. This allele form the 2 gametes, this again allele tallness form the 2 gametes, even this short recessive allele formed again 2 gametes, even this short allele again formed 2 gametes, but this is the gamete formation by this will not have implications on this, or gamete formation by this will not have the implications on this. So this is nothing but called the law of independent assortment. So today's last subtopic we are going to study about the monohybrid and dihybrid and trihybrid cross. Cross means just pollination between the two plants of contrasting characters. Now here mono means just one, so definitely I will just select only one characteristics or contrasting characteristic. This is a tall and short. Now if these two plants, if there is a hybridization between them or pollination between them, it is cell, then what will the outcomes, or this can be given by the Punnett square. Now this is I will write this uh, alleles T and T, and this will be TT. Next, this T and this small t will form capital T, small t. This capital T and small t will form TT. Next is TT, and this is. So now you will find this very important thing here is just how to write the phenotypic ratio and the genotypic ratio. Now if phenotype ratio is just external appearance, I told already, I explained about it. Now this capital T, capital T, capital T, capital T, all will have the dominant character. So definitely the dominant character will exist after the F1 generation, first filial generation. That means all the plants will be tall. So I can write phenotypic ratio as 4:0. Now with respect to genotype ratio, now we will talk about whether it is heterozygous or homozygous. That is what genotype is. Now this is again heterozygous. This is heterozygous because it has different alleles. Heterozygous, different alleles, heterozygous and the heterozygous. That means again genotypic ratio will be 4:0. That means all are heterozygous. So in F1 filial generation, we have phenotypic ratio for monohybrid is 4:0, and genotypic ratio for monohybrid is 4:0. Now I will do the selfing between the plants in the F1 generation, that is starting of F2 generation actually. So it is capital T small t, capital T small t. That means this and this plant I am selfing it. Selfing, I told you, it is a pollination or hybridization between the plants of same generation. So this I am doing it here. So what are the gametes possibilities are here? It will be T, 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 T. So again same thing I will write down. This capital T small t here, and again capital T small t here. So this capital T and the capital T will form capital TT, capital T small t. Small t capital T means capital T small t, and the small t small t. Now this is at the end of F2 filial generation. Now this is here, this capital T is homozygous and it's a tall because both are same alleles. Now here heterozygous, they are different, but dominant character exists, so that's capital T, so it will be tall. So it is a heterozygous tall, it is a homozygous tall. This is capital T small t, it is again a heterozygous tall, small t. Now this one is homozygous DOF. That means homozygous, why? Because they are having the both same alleles, and they are the small t, and small t is nothing but the DOF notation. So phenotype ratio, there are the three talls, 1, 2, 3. In phenotype, it doesn't matter whether it is heterozygous or homozygous, just we have to check whether the dominant characteristics. So external appearance, this all will be tall. So this plant will be tall, this P plant will be tall, is again tall, but this will be short. So this is the homozygous will come, this is heterozygous, this is again heterozygous, this is homozygous short. So now while writing the phenotypic ratio, external appearance, 3 are tall, 1 is short, so I can write 3:1. And genotype ratio, in genotype ratio we have to consider the homozygous and heterozygous. Now here this is homozygous tall, so I can write as a 1. Next is, this is heterozygous tall, so 2, and 1 is homozygous, which is short, so it is a 1. So there are 3 possibilities: homozygous tall, heterozygous tall, and homozygous short. For all the 3 we have to write the 1:2, 2:1, because homozygous tall is 1, heterozygous tall is 2, and the homozygous short is 1. So genotype ratio is 1:2, 2:1, and the phenotype ratio is 3:1 for F2 filial generation for the monohybrid, for the monohybrid cross. Now this is about the monohybrid cross. Next is dihybrid. So dihybrid means, the name itself indicates is two contrasting characters we have to take. So I have taken the height of the plant, that's capital T dominant, and the color of the flower, that's purple, so capital P I noted here, and it is pollinated or hybridized with small t, that's recessive characteristics, and then small p, that's the white color of the flower. Now again, the two characteristics hybridization or pollination, that is called dihybrid cross. So what are the possible gametes? This T and this capital P, so will form TP. Again this T and this capital P, TP. This capital T, capital P, again TP, and capital T, capital P, TP. So this is all I will write here. Then small t, small p, small t, small p. So like 4, so I'll write 4 here. Now we have to write down, this is dihybrid, so 4 letters will come here. Capital T, small t, that is what here. Capital P, small p here. Now here again capital T, small t, and capital P, small p. So this all will be, everything will be same, capital T, small t, capital P, small p. So definitely this is the F1 filial generations outcomes. So I can say from the Punnett square, in F1 generation, phenotypic ratio, because the elements are total 16 here, so phenotypic ratio, all are heterozygous tall, so phenotypic ratio is 16:0, and genotype ratio, all are heterozygous, so it is again a 16:0. So phenotypic ratio, all are tall and purple, all 16 plants, so 16:0. Genotype ratio, all are heterozygous, so it is will be 16:0. So you can easily predict it by just writing the gametes and then just pollinating them, or hybridization, just write down in the Punnett square, and easily we can say the phenotype ratio and the genotype ratio. Phenotype ratio, it is a heterozygous, we know, but it's a tall and purple because tall is dominant and capital P is purple. So all will be TTPP, small and the capital, so it will be tall and purple. And genotype ratio, it's all are heterozygous because T and small t, so different alleles, so heterozygous, capital P, small p, heterozygous. All 16s are heterozygous, so genotypic ratio also 16:0. Now this is the F1 generation we studied. Now we'll study about the F2 generation for a dihybrid cross. I told you we have to do the selfing. Selfing means just crossing the plants from the first filial generations. So this is capital T, small t, capital P, small p, again capital T, small t, capital P, small p. So again same thing, the possibilities here for forming gamete, this capital T and capital P, so this is gamete. Capital T and small p, so this is gamete. Small t and capital P, so this is 1, and small t and small p. So this TTPP, first is TP right down here, the one is capital T, small p, next is small t, capital P is here, next is small t, small p. So, and here capital T, capital P, capital T, small p, small t, capital P, and small t, small p, you can write down here all the gametes, and just cross it. So capital T, capital P, capital T, and this capital T, capital P, and capital P. So accordingly you have to write down all the 16 elements, write down properly all the 16 possibilities, and then you check. Now what are the possibilities here? One possibility will be plants will be tall and purple. Tall and purple means it should consist of capital T and capital P. So you have to count it, which are the plants possibilities consist of capital T and capital P. If it consists, then it will be the tall and purple. Next is possibilities, it may be tall, but the flowers will be white. So capital T and small p. So you have to consider which consists of capital T and the small p. Next is possibility, the tree may be short, but the flower will be purple. So this is also possibility, that will small t and the capital P. And next will be the short, both this even capital T and small p, both are recessive characters. So this also you have to see which are the how many elements having these properties. And if you write down properly, then you will get nine possibilities will be tall and purple, 3 possibilities will be tall and white, 3 possibilities will be short and purple, and 1 possibility will be short and white. So we can write 9:2, 3:2, 3:1 as the phenotypic ratio for the F2 generation for dihybrid cross. Thank you. And the last topic is tri-hybrid cross. This is again tri, so definitely I will take three contrasting characters, and the characters what I've chosen is tall, purple, and axial. So tall is capital TT, purple is capital PP, and axial is capital AA. This is the flower position, color of the flower, and the height of the plant. Next here is short, white, and terminal. So short is small TT, white is small PP, and terminal is AA. The position, how it look like for terminal and axial, I have just I have explained in the slide where I explained all the 7 characteristics what Mendel had considered. So please see it, refer that chart. Now here I will say in this 3 contrasting characters what are the possibilities of gamete formation. So there are 8 gametes will be formed in this recessive, so 8 gametes will be formed. So now writing 8 gametes vertically and horizontally, and writing 8 gametes vertically, it's 64 elements we have to write down. So please write down and check the 64 table. Same thing you have to write down TPA. So what are the gametes will form? These gamete possibilities I have given here. The 8 gametes, which are the 8 gametes will form, I have given the possibilities here. So just look at the possibilities of the gametes will be formed in the trihybrid. So this will be 8. So you'll write the 8 in the horizontal manner, 8 in the vertical manner, and just cross these gametes. You will get the outcome result, what is going to come. And then if after writing, we can observe the phenotypic ratio was 64:0 because this is F1. I'm talking about F1, first filial generation. In F1, as we know, dominant traits of the offsprings will always exist. So dominant was tall, purple, and axial. So it is 64:0 because this recessive there is no chance of coming into it. Genotypic ratio definitely dominant again. So it may be heterozygous, but all will be the dominant. That's tall, purple, and axial, and now heterozygous. So genotypic ratio also will be 64:0. Now, so we got the phenotypic and genotypic ratio for the first filial generation. In the F2, second filial generations, we know that for the second filial generation, we'll do the selfing between the offsprings, what we observed at the end of the first filial generation. This we have to now cross. So the possibility of forming the gametes of this one is capital T and capital P, capital A, because 3 we have to consider. Next is capital T, capital P, and small a. Next is capital T, small p, and capital A. So like this we have to form the gametes, all 8 possible gametes. If they ask what are the possible gametes we can form from this trihybrid cross, there is 8 gametes. That's correct. But that is a possible gametes. If they ask you what is types of gametes, types of gametes are different. Types of gametes means we have to write only for the one capital T. So here at TPA, T small p capital A, capital T capital P and small p, capital T small p and small a. These are the types of gametes will be formed, and the complete possibilities will be the 8. So please remember this one. Now next, this is for finding genotypic ratio by at the end of the F2 generation in trihybrid cross is a very hectic task. But for MCQs they may ask you what is genotypic ratio for the second filial generation for the trihybrid cross, and in the exam we don't have much time. Hardly you get the 2 minutes or 1.5 minutes to solve this question. You cannot just make Punnett square of the 64 elements and take chance. It will take near about 15 to 20 minutes, and that's very costlier. So we'll discuss how mathematically we can solve this one. It's quite easy. So now here, if you see, these are methods I have given. First I will write down how to find the phenotypic ratio, and then we'll see what are the method to use for finding the genotypic ratio. Now general formula to find the phenotype ratio for all the crosses, whether it is a monohybrid cross, whether it is a dihybrid cross, or whether it is a trihybrid cross. Phenotypic ratio formula is 3:1 to the power of n. See again, 3s to 1 power n. We have to always remember this formula. Don't forget it because they have asked the questions on this. So 3s to 1 to the power of n. Now the question is what this n indicates. It indicates the selection of contrasting characters. So, or selection of number of traits. So definitely this for monohybrid, the characteristics what I had chosen is just 1. So I can say 3s to 1 to the power of 1 for monohybrid, and this is 1, so nothing is going to change. So phenotypic ratio is 3s to 1. You can compare this answer with how we find out for the by using the Punnett square. You can just compare it with. Next is dihybrid cross. Dihybrid cross means two characters we have to choose it, and that's 3s to 1. So same as it is will come. N value will be 2 because it is a dihybrid. Now this means multiply 3s to 1 into 3s to 1. It is not normal multiplication. Remember again, 2 times we have to 3s to 1 into 3s to 1. Then what we have to do it? How? See here. This 3 into 3, that's 9. 3 into 1, that's 3. So for 3 is done. Now again with 1 we have to multiply. 1 into 3 is 3. 1 into 1 is 1. So it is 9s2, 3s2, 3s2, 1. Again I will explain. 3s to 3, that's 9. 3s to 1, that's 3. 1 into 3, that's 3. 1 into 1, that's 1. So 9s2, 3s2, 3s2, 1. This is what the phenotypic ratio, and you can even compare our answer with respect to the answer we can find by the Punnett square. So next is trihybrid. So definitely 3 times we have to 3s to 1 to the power of 3 will come. Now 3 times 3s to 1, 3s to 1, 3s to 1. But 3s to 1 into 3s to 1 answer is this, because for dihybrid already we found out. So this into 3s to 1 we can say. Same thing. 3 into 9 is 27. 3 into 3 is 9. Again 3 into 3 is 9. 3 into 1 is 3. Again 1 into 9, 1 into 3, 1 into 3, 1 into 1. So this is what we got the phenotypic ratio for the dihybrid cross. And now we can interchange it, take all the 9s together, all the 3s together, and 1. It looks good actually. So even you can interchange it. So I hope you understood the phenotype ratio, how to find out for the mono, di and trihybrid cross between the plants. Next we'll study how we can find the genotype ratio. Just we have to remember the genotype ratio basic formula is 1s to 2s to 1 to the power of n. N again same number of traits or contrasting characters we have to select for the particular cross. For monohybrid we have to select 1, so 1 to 1 to the power of 1, and this is again you can check the answer, recheck the answer by using the Punnett square and the method of by this formula. So same answer we got, 1s to 2s to 1. Next is dihybrid, so by 2 times this into this, again 1 into 1, 1 into 2, 1 into 1, then 2 into 1, 2, 2 2s are 4, and 2 into 1, again 1 into 1, 1 into 2, 1 into 1. This is exactly 9 elements will come. So like this we can find out. Now trihybrid, 1s to 2s to 1 to the power of 3. Now for the 2 you already found out, so write down the 2 and into 1 into 2 into 1. Again you have to multiply this 1 into all, 2 into all, and 1 into all. Please find out. So 1 2 3 4 5 6 7 8 9 9 3s are 27 elements you will get, because if you multiply with 1 you will get 1 2 1 2 4 2 1 2 1. If you multiply with 2 you will get 2 4 2 4 8 4 2 4 1. If you multiply with 1 1 2 1 2 4 2 1 2 1 will come. That means for every element you will get the 9 proportional ratios. So total 27 elements you will get. I hope you understood the mono, di and trihybrid crosses for the according to Mendelism, and how we can find by using the Punnett square and how we can find by using the formulas. So this was today's lecture. Please read the NCERT books for more further information. If any doubts are coming, don't hesitate to ask me. I'm ready to give the answer at any time. Please message me if doubts are there. Even you can just message me which topic you want to repeat it again. This is very important topic, so please revise it very well. Get the as information is possible, gain the knowledge about this topic. It is extremely important. I hope you are good at your place and studying very well. Thank you. The next lecture will post it on day after tomorrow. Till then,